Well, here we are. We are doing the Mitosis and Meiosis cell division lab. And the nice thing is that this lab is mostly modeling. So we don't need our safety glasses and our gloves and our aprons. So, a couple of things I want to talk about. Both mitosis and meiosis are types of cell division. Mitosis will be the cell division where one cell makes two genetically identical cells, and this is really happening for eukaryotic cells. So if my original cell had 10 chromosomes, at the end of mitosis I will have 2 cells, and each cell will have 10 chromosomes. We need both of those cells at the end of mitosis to have every single piece of DNA, every gene, because those cells are going to be replacing damaged cells, replacing old cells, or potentially growing the organism. We need those cells to serve the same function in the tissue and the organ. Now, meiosis is going to be that really super specialized type of cell division that will happen just for reproduction. So we've got specialized cells that go through meiosis. They're going to produce the gamete cells, either the sperm or the egg. With meiosis, our cell is going to have to go through 2 rounds of cell division. So our cell will go through prophase 1, metaphase 1, anaphase 1, telophase 1 of meiosis 1, and then have to go through prophase 2, metaphase 2, anaphase 2, telophase 2 to make 4 genetically non-identical cells to make the gamete cells, either the sperm or the egg. Now, the reason why our specialized cells are going to go through meiosis is because we need to decrease the amount of genetic information. We're going to cut the amount of DNA, the amount of chromosomes in half. So if our original cell started with 10 chromosomes, at the end of meiosis, I will have 4 cells that are haploid. They have half the amount of DNA. Each cell will have just 5 pieces. Now, when the sperm and the egg come together, our new diploid zygote, our kid, our next generation, whatever you want to call it, will have 10 pieces of DNA, just like that original cell, just like the parents. And so across generations, we're going to see the amount of DNA stays constant. So if I looked inside of your cells would hopefully have 46 individual pieces of DNA. Your parents' cells hopefully have 46 individual pieces of DNA. Your grandparents, your great-great-great-grandparents, we could keep going. The reason why we all have the same amount of DNA from one generation to the next is because of meiosis. So today, we're going to start with the simpler process, mitosis, where one cell makes 2 genetically identical cells, and then we'll work our way into that more complicated meiosis modeling. Make sure you get your chromosome kit. And one of the things you may notice about your chromosome kit is that some of your chromosomes may be missing little beads. Now we can think of each of these little beads as being individual genes. So this is not a good situation to start with. Take a look at the chromosome chart in Hands-On Lab. In the chromosome chart, it will tell you how many beads you need on each of your arms. So the big white piece here is the centromere, and some of our chromosomes will have 4 beads per arm. So I've got 4 beads on this arm, and then I've got 4 beads on this arm. Now other chromosomes are going to ask you to have 3 beads. These pop apart fairly easily, so do be careful when you're using them. It's easy to lose beads, and you don't want to lose your beads for the modeling. So here I have made a chromosome that has 3 beads on each arm, and then I have my big centromere in the middle. A centromere is a magnet, and as I was showing you earlier, they do all stay locked together. So it's going to help us with our modeling today. A couple of things you're going to notice. In the Hands-On Lab, they're going to talk about a phase called prometaphase. In the Bio 103 class, we don't make this distinction. When we look at either mitosis or meiosis, we're going to separate it into the 4 main phases. prophase, metaphase, anaphase, and telophase. So pro-metaphase generally gets lumped in with prophase. Make sure that your labels for your pictures are very clear and easy for me to read and understand. If you have any questions, always email me. My last big piece of advice, I posted 2 videos that I use for my Bio 103 class, where what I'm doing is I'm modeling the process of mitosis and modeling the process of meiosis using socks. This can be a really good review for what you would expect to see in each phase. I also use the terminology, so maybe a review of the terminology will help you to understand what's needed for this lab. I look forward to your lab reports, and as always, you know how to get a hold of me. I hope you're holding in there. We're almost towards the end of the semester.